All right, thank you very much, Byron. Appreciate it. It's really nice to see everybody again, and really looking forward to discussing how NGS can be used for snow leopards and just other potential ways that we can think about trying to incorporate it into studying this remarkable species and just in general understanding wildlife populations, adaptation. I'll try to keep it focused to the sort of like the main techniques that are potentially transferable to non-invasive genetics, and then I'll highlight some of the work that we're working on right now and how we're trying to use it to understand how snow leopards have adapted to hypoxia. I also want to discuss some issues with the challenges of trying to incorporate NGS into wildlife studies, particularly, and some of the issues that come about when you try to do that. And so again, I hope this stimulates a discussion and ways we can think about trying to really leverage these techniques to improve our look at the populations, not just the snow leopards, other species, and specifically improving conservation management, and then just understanding the whole ecosystem there. So next generation sequencing has been used about the last decade heavily to understand a lot of different things. So it's great for characterizing not only, the specific individual genetic diversity that you see in a particular population, but whole species as a whole, comparing and looking at how different species and communities have radiated, evolved, adapted to different environments. And what's great is that NGS has allowed us to, instead of looking at a single gene or a couple genes, couple markers, to look at the whole genome perspective, right? And so we get a much better resolution of understanding the demography and history of populations and species. Again, how these species have evolved and diversified. And then also now we're starting to be able to have the technology to really look at what is the genetic basis for different phenotypes, for example, the pathogenesis and the progression of different diseases, epidemiology, how are these diseases and viruses and things that could be affecting populations distributed. Now, these techniques have, of course, been developed and really advanced in model organisms, such as the picture here, the stickleback. So some of the techniques that you probably have heard of, like rad-seq, which I won't discuss today, just because there's a limited things I can cover. But rad-seq was really developed and used on the stickleback to try to understand the evolution of some of these different phenotypes and different isolated populations, right? Another photo I have in there in humans, there's these rare diseases that are heritable. Researchers have been able to, just through sequencing the entire exomes of a couple individuals, identify the genetic basis for these diseases. And then there's been, really interesting things looking at the tree that I have here. Let's see, can you guys see my laser pointer? There we go. This here, it shows the clustering of the Plasmodium parasite in different sample from different countries. And with these parasites, there's actually populations in Southeast Asia that are resistant to the malaria drug artemisinin, right? And so basically trying to understand how these populations are connected and how potentially the resistance to this important drug can be spread throughout these populations. All these things basically benefit from incorporating NGS because, again, we can start looking at a whole genome level, right? And then it expands what kind of questions we can ask and address. And so I want to highlight first, there's two main kinds of molecular markers that people use for population genetics, right? And so the first is microsatellites. And again, microsatellites are simple tandem repeats. So there's a sequence of a microsatellite depicted here. In this case, it's a repeat of ATAG. And basically the alleles or the variants you examine are just simply insertions or deletions of a particular number of repeats. One, let's say, repeat unit in this particular microsatellite would change the size of that particular region of the DNA by 4 base pairs, right? And so basically by looking at the number of alleles that are produced by mutations of these insertions, deletions of repeats, you can track the variation in a population, and you can use it to, for example, identify different individuals, because again, the combination of the unique alleles they're going to have is going to be able to give you individual ID. It acts like a genetic fingerprint. And then how these alleles are distributed, over time and space, gives you a lot of information on the population dynamics, the connectivity, fluctuations in populations, and so forth. And so primarily because they're so variable, and you can have up to 5 to 15 alleles per single microsatellite, there's a lot of information in those microsats. You don't have to genotype that many to get a good handle on the genetic diversity to some extent, right? By many, I mean like, it should be between 10 to 30 or 40, up to 40 in some cases if you're looking at fine structure. But these things could be genotyped with a, standard Sanger sequencing instrument. Now, that other type of variation is a single nucleotide polymorphism, where you have a substitution that changes a nucleotide. And for example, in this case, the allele A has an A at position 299 to a C, same position in allele C. Now, typically, single nucleotide polymorphisms, you have to sequence to figure out what nucleotide's there. And because, sequencing is more expensive than genotyping, also there's only 2 alleles per locus, so there's less information per marker. Typically, you have to genotype more SNPs to basically get the same information that you get from microsatellites. So because of this, a lot of population studies typically focused on microsatellites, right? But now, because there's these next generation sequencing instruments, and the primary dominant player in this is Illumina, they can basically sequence millions of DNA segments at a time, or molecular markers at a time, and allows you to efficiently get SNP information, or just consensus sequence, for a vast amount of markers at a single time, right? And basically, this is basically what happens is the sequencing on an Illumina instrument is performed on a little glass slide, basically, that has these oligonucleotides attached to it. And then through bridge amplification, you amplify this cluster from originally source as a single DNA fragment that comes from your sample. And basically, based on the nucleotide that is incorporated during the sequencing reaction, there's an image taken of the slide. And each of these little dots, these little clusters, represents basically a single nucleotide that has been added to a DNA fragment that's being sequenced, basically, right? And so from what color of that spot there is, you can read the sequence of that DNA molecule. And because this can happen, in this really high throughput, you can sequence up to 6 billion, actually this is GB as billion, so 6 trillion base pairs at a time with some of these instruments, right? So this sequencing technology was developed by a company called Solexa in 1998. Illumina purchased them in 2007, and basically, as they have developed these instruments, they've came up with different ones that have different capacity. And so again, they can sequence anywhere from maybe 300 million base pairs at a time to up to 6 trillion base pairs at a time. The read lengths have changed from vary from 50 base pairs to up to 500 base pairs. And so I'm going to just give you a little brief overview of how it works. So basically, you shear your DNA, you ligate on these adapters, or you can incorporate adapters through a PCR reaction. And then basically your DNA fragments that are depicted in this gray, they have these matching adapters that can allow a single DNA fragment to ligate to that flow cell, right? So this is, again, this thing down here is just basically a glass slide, like a microscope slide. And then through a process called bridge amplification, that single DNA molecule that's bound to that flow cell is copied and amplified. It's very similar to a PCR, but it's just that PCR is just fixed onto that point of that slide where that DNA was attached. And then it generates a amplified cluster that, again, is basically a copy of that original DNA fragment that that was attached to that flow cell, right? And so in this particular picture, each of these 4 different clusters came from a single DNA molecule, right? Now, as the sequencing reaction progresses, when that fragment is being sequenced, basically there's a nucleotide that is incorporated in that specific spot that, for each of those DNA fragments that's being sequenced, and basically that color of that particular spot reflects which nucleotide was incorporated, right? So in this particular sense, in this cycle, a G was incorporated in this spot, a T in this spot, a C in this spot, and A in this spot, right? And if you repeat this over many, many, many cycles, you can basically get the sequence of the DNA fragment that was used to produce each one of these little clusters, right? So it's really remarkable. And what's really crazy is that, it's on a little MiSeq slide. I mean, that thing measures just maybe 2-3 centimeters by a centimeter. You can generate 25 million clusters. And so you're sequencing 25 million DNA fragments at a time, right? And so because each of these spots is really equivalent to a single Sanger capillary, you can sequence on a MiSeq 25 million fragments compared to a maximum of 96 capillaries on the big ABI instrument 3730, right? And so in comparison to generating around the max of 67,000 base pairs in a single Sanger sequencing ABI 3730 run, you can produce up to 15 billion pairs of sequence on a MiSeq, right? So just to show you how vast difference this is and how scaled up this is, if you had a single Sanger sequencing instrument that you ran every single day without a break, it would take you approximately 300 years to sequence the same amount as a MiSeq that it takes just 3 days to do. That is how phenomenally, the scaled up these things are, right? And the MiSeq is actually one of their smallest instruments. It just does a fraction of sequence for the large instrument. Now, what's really great about Illumina sequencing as well is that it allows you to multiplex the samples, right? And so basically during the production of these templates that you're going to sequence on the instrument, you can also have, let's say, 2 samples, and it can be, currently you can fairly easily multiplex up to 384 samples and sequence them at once, okay? And basically each of these samples can have a different barcode, and barcode is just a unique nucleotide sequence at the beginning that you can resolve at the end, whether this DNA sequence read that's generated comes from sample 1 or if it comes from sample 2, right? And so this again allows you to basically be able to sequence, for, let's say, a sequencing run can cost about $1,500. So if you could only do 1 sample on that, it would be too expensive, right? But because you can sequence up to, let's say, easily a couple hundred samples at a time, all of a sudden that cost of the sequencing drops to just $15, $20 per sample, right? So it makes it more reasonable. And then what you can do, the 2 primary ways that sequencing data is used to generate something that's usable for population genetics is either you create a consensus sequence from the reads. And so basically what you're going to do is, so if this is your, let's say the sequence you're trying to get, the reference sequence of the DNA, let's say COX-1 or cytochrome B, for what Charlotte did for the diet analysis. And you have these short reads that are mapping to that particular sequence, right? And so in this particular case, it's showing if you had, for example, you had an entire gene and then you sequence multiple fragments that are coming from that same large gene, right? And so you map those reads down here to the actual reference sequence, and how many reads are mapping in a particular spot reflects the coverage or the sequencing depth, right? And if you basically stack all those sequences that come from the same spot on top of each other, then you can basically extract what's referred to as the consensus sequence down here, right? So this is the consensus sequence that basically matches and is for the same spot as your reference, but now that consensus is what's been generated for your actual sample, right? In some cases, it's going to be identical, and there's going to be places where it's different from the reference. Now, that's what you can use, that consensus sequence, is then you can use and do things like what Charlotte did for her data analysis. You can then search that against larger reference database collection and see, for example, which species that consensus sequence matches, and most likely was present inside that DNA sample, inside that scat. Another way that you can use it is you can do what's referred to as genotyping by sequencing, right? And so this is used in population genomics, functional genomics. Basically what you're doing is if you have the reference sequence that's depicted up top, and you have a single nucleotide polymorphism marker right here, it's highlighted in red, and you map the sequences, the reads that match that particular region, right? You basically can see how many of those reads have a different nucleotide at that marker, right? And in this particular case, we have 5 reads that have an A and are different from the reference. And then we have 6 reads that have a G or are the same, right? And so there's 5, about half the reads support an A, half the reads support a G. This is a diploid individual, right? So you'd expect that individual is heterozygote in that position, and the genotype for that particular SNP marker is AG, right? Now think about it. Now we can expand, because we can sequence, millions and millions of fragments at a time. It allows us to now scale this up. And, using traditional Sanger sequencing, for example, people used to genotype 30 to 40 SNPs or maybe 100 SNPs. Now we can genotype thousands of SNPs at a time, potentially. Okay, and so one thing I do want to point out that the Sanger sequencing instruments, that those first generation sequencers, they're still good because they have a higher accuracy. They're cheaper, so the cost of running the instrument is lower. If you're only doing a couple samples, a couple genes at a time, it's still, and you have a specific sequence, sometimes it's advantageous to still use the Sanger sequencer, right? For microsatellite genotyping, microsatellite genotypes are, still really good markers for individual IDs, for population genetics, and so they're still useful for these types of studies, right? So for microsatellites, Sanger sequencing is still great for genotyping those, right? Now, Illumina and next-gen sequencing, it's when you start getting into genome assembly, transcriptome, differential expression, metagenomes, that's where Illumina really shines, right? In terms of applications for snow leopard, when you're doing, for example, 16S, 12S, or COX-1 barcoding or Site B, where you're trying to identify a mix of unknown samples, with a particular sequence, right? This is ideal for that, right? So again, the technique you're doing is mapping reads, extracting a consensus for different samples. For phylogenetics, same thing. Now, instead of just, when we did the original phylogeography paper on the snow leopards, we just sequenced, some short fragments in mitochondrial DNA. There was no variation there that we observed. However, with these NGS methods, we're trying now to basically sequence the entire mitochondrial genome. We can do that now with NGS, right? For population genomics, genotyping by sequencing, again, instead of just looking at 30 microsatellites, we have the potential to look at hundreds to thousands of SNPs using methods such as RAD-seq, target selection, and amplicons. And functional genomics. So basically now we can start to look at things that are related to the biology of the snow leopards, right? Adaptation to high altitude, behavior, things like that, disease susceptibility, these sorts of things. The questions that we can now ask in a sort of conservation framework, ecological framework, greatly increases because, again, we can explore things from a whole genome perspective, right? Now, I'm going to discuss basically 2 primary ways where you can save money and allow you to do these wildlife studies with NGS, because again, it's too expensive to sequence whole genomes for a particular population, right? And so this figure up here kind of depicts the trade-off. So if you try to use NGS to look at some sort of question, you basically, and for a fixed budget, you have 2 options, right? You either sequence a large proportion of the genome and a small number of individuals, or there's the opposite where you sequence a small fraction of the genome, but a lot of individuals, right? And so low coverage whole genome sequencing is one way where you can get sequence from, roughly the whole genome, but you're only sequencing them at very light coverage, right? So you don't have a lot of reads stacking on top of the same location, right? But you're only doing this for a small number of individuals, right? And so this is where you sequence anywhere from 0.5 to 5x for maybe a couple dozen individuals. And you can also pull those into library. In some cases, that's effective. In other cases, you can also do what's referred to as reduced representation sequencing. So this is where you target a specific part of a genome. You do this targeted enrichment where you select the DNA fragments that you want to sequence either through PCR amplification or hybridization, and then you sequence them at higher depth for many individuals, right? Tens to hundreds of individuals. But you're only sequencing a small fraction of the genome. But you're specifically sequencing certain areas. Other possibility is to do restriction enzyme digestion and sequencing. That's what's referred to as RAD-seq, right? Double RAD-seq. In these areas, you're sequencing a small fraction of the genome at high percentage. It's fairly random as to what sites you're sequencing, and you can do this for hundreds of individuals, right? So again, these are two slightly different strategies that allow you to apply NGS in a larger scale for wildlife studies, right? Whether you're going to do low coverage sequencing, where again, you kind of get a snapshot of the whole genome, but at whole coverage, or RAD-seq. So basically RAD-seq, you're sequencing off restriction enzyme sites. It's sort of a random set among the different chromosomes versus enrichment, where you're sequencing a specific region and you're resequencing it, everybody. There's lots of different things to consider. Things like how much of the genome do you want to target? Are the reads going to be mapping to a specific location? Are there specific markers that you want to sequence? Will the coverage vary between these different specific targets of markers? How are you going to be able to generate the data reproducibly across? Are you trying to basically have data that's comparable across different study sites, across different research groups? Even logistical stuff like what's your budget? How many samples do you need to sequence? How many samples do you have funding to sequence? How easy it is to generate those DNA libraries. And then also the other really important aspect is the DNA. So we talked a lot about how DNA is degraded, low quantity usually in the scat. And so you have to make sure that the DNA the method you're going to use is actually possible given the limitations there are with the DNA quality and scat, right? So again, low coverage sequencing, you're getting potentially reads from entire chromosomes at light coverage. One advantage of doing this is that you basically can pool all the samples together, sequence one library, and basically because the allele frequencies are going to be, you're going to be able to basically get a good estimate of them from the amount of reads that are mapping to each of the different alleles. You're basically able to fairly efficiently get a kind of a snapshot of the whole genome within a population, right? However, the disadvantage is that you lose individual data, right? So basically you're treating populations as a pool. Now in some type of analyses, like in some approaches to landscape genetics, that's okay. In other cases, you need individual information, right? Way to get the individual information is, again, to do some kind of target enrichment. One common way to do this is through a PCR amplification, where you design primers for specific molecular markers that you want to genotype, and you basically then multiplex it at this larger scale, right? And so in this particular case, you can easily generate data for over 25,000 base pairs total sequence for maybe 96, a couple hundred samples, or up to a couple 100,000 base pairs of sequence for markers from maybe a couple dozen samples, right? Of course, it can be further scaled up, but I'm just giving kind of general range that's reasonable cost. And there's basically three different strategies. Either you can
just simply use the PCR, right? So the PCR is a very robust way of increasing the copies of a certain sequence target. The basic bottleneck, though, is just primer design and trying to optimize PCR, especially when you're trying to multiplex a lot of different primers, right? And so there's three really general ways that you can apply the PCR towards Illumina libraries and make Illumina libraries. The first one is that you can do a uniplex, and so basically you can design primers along a larger DNA segment. So for example, up to 20 kb is pretty easy to amplify, 20,000 base pairs. And then these larger amplicons that are going to have the markers that you want to sequence, so you can genotype them by sequencing, can then simply be input into the DNA library preparation methods that you would normally use for generating genome libraries, right? The other two strategies, however, are more amendable for DNA that is degraded or of low quantity like you have in SCAT, right? And so basically the way it works is that you just design primers that are adjacent or on either side of the specific SNPs or the markers that you want to sequence, right? And you can do a kind of a low throughput multiplex where you can design, for example, let's say 10 primer pairs to amplify 10 different SNPs. And then potentially you can do a couple different multiplex reactions for the same sample, right? So let's say if you do 10 multiplex reactions with 10 different primer pairs, you can basically amplify up to 100 different SNPs and then sequence them, right, by pulling them together. And typically if you try to, keep those lengths of those amplicons below 200 base pairs, you can fairly easily amplify them in SCAT samples, right? Now, this is taken one step further where there's companies, so for example, one proprietary method, Rainstorm, is, or basically it's like a product that's marketed by a company that helps to work with you by designing up to 4,000 amplicons that can be basically multiplexed at one time in this massive reaction, right? Now, of course, the more amplicons that you try to amplify together, the harder it is to optimize it, right? But it's a good approach. Another way that you can create a target enrichment library is just through hybridization. So basically what you're doing is you can design these oligonucleotide probes, right? And so these probes can be either designed by PCR amplifying that target in a good quality sample, let's say from a blood sample, or you can actually, again, pay a company to design and make oligonucleotides, and then basically you label these, what are going to be the probes, with biotin, and then you take your DNA sample that can be degraded, so from a SCAT extraction, for example, and you hybridize your DNA sample to these oligonucleotide probes that are labeled with biotin, and then you bind those together to a magnetic bead that has streptavidin attached to it, right? The streptavidin and the biotin closely go together, and basically they stick together. You can use those magnetic beads to then isolate the targeted DNA that's hybridized to those probes and wash off all the other DNA that you don't want to sequence, right? And all the other junk. And then you release the DNA and you sequence it, right? And so this method is also very amendable to degraded DNA samples. There's actually a couple of really nice papers that have optimized this method for use in museum specimens. And again, museum specimens often are degraded just like SCAT samples. Now, there's kind of this trade-off between PCR and hybridization, right? So basically, if you want to sequence, smaller amount of markers for more samples, probably the PCR approach is more of the way to go. If you want to sequence more markers for fewer samples, the target enrichment using hybridization would probably be more efficient to do it. But both of those strategies would work well. I'm going to talk about an example of how we use NGS to better understand snow leopards, and specifically focusing on their adaptation to high altitude, right? So my lab for a long time has been doing non-invasive surveys. We've been working with groups to look at phylogeography, structure, diet. That's one of the primary focuses of my PhD student, Charlotte Hacker, as she talked about it during the workshop. And then recently we've got into genomics, where we're sequencing the genomes of different individuals, particularly from the captive zoo population. And then we even developed some models where we're using cell culture to look at gene expression in white blood cells from snow leopards to see whether or not they have different responses to hypoxia, potentially, than some of the other cats that they're related to. Now, why would we look at this? So think about this. Snow leopards occur in the highest altitude region in the world, right? And so a lot of these animals have to survive really low oxygen levels, and so they're adapted for that low oxygen hypoxic environment, right? Well, when you think about it, when we have these anthropogenic effects like climate change, these habitat shifts, other pressures from humans, like development usually occurs in lower altitudes, right? There's basically different levels of range degradation, grazing patterns, all these things. In general, species tends to get pushed up higher into the mountains, right? But the question is there some sort of upper limit to how far up in elevation snow leopards can go and other species can go? Now, about 6 or 7 years ago, there was a really nice study that was published by Chow et al. That, again, used next generation sequencing, and they characterized these two genes that had unique alleles, mutations that became fixed, or at least what it looked like at the time, both of them became fixed in snow leopards. One is EGLN1 and one is EPAS1. Both of these genes are transcription factors that regulate a cell's response to hypoxia, right? And so they do everything when your oxygen levels drop. These two central genes basically stimulate the response of your cells to, for example, increase your ventilation rate. If it's increase red blood cell production, if it's hypoxia that occurs over a longer period of time, there's other genes that are turned on that basically stimulate angiogenesis or production of capillaries and other things. And so again, because these two genes are so important to the response to low levels of oxygen, they are presumably adaptive in snow leopards. Now, a couple years ago, we published the first phylogeographic study of snow leopards. And what we found was that snow leopards are structured into these two fairly distinct genetic clusters, right? One cluster is in Mongolia, the northern cluster. There's a cluster in the central part of the range, in sort of the central eastern Tibetan Plateau. And then there's another cluster in the western part of the range into the Karakoram, Kyrgyzstan, and Tian Shan mountains. Now, because of the distinction of these three genetic clusters, we proposed that they were three different subspecies. Now, of course, this was controversial to some extent because the snow leopards are monotypic, and other people argued that there really wasn't enough evidence to evaluate them as subspecies, right? Now, we did reply to this. We, again, reiterated some of our arguments, why we think that is a valid classification. However, really, whether it's a subspecies, ESU, it's sort of an academic exercise, right, to some extent. It does have some important consequences for conservation, and which is one of the reasons why we think that the taxonomy of snow leopards is basically more consistent as breaking down as subspecies if we look at the genetics and the biogeography. However, ultimately, whether or not they're categorized as subspecies or monotypic, really what we need is more information. And so we need more data to examine the genome-wide patterns of structure in this particular region, right? And so if we genotype SNPs across the whole genome, are we going to have patterns that are supporting one versus the other, right? And the only way we're going to know is by doing more in-depth genotyping and sequencing, right? The other important question is, now that we have these potentially three genetic clusters or different genetic populations or groups, again, whatever you want to call them, ESUs or subspecies, are there actually functional differences between these different areas? And then finally, now that we've discussed that snow leopards have these high altitude adaptations potentially in these two genes that respond to hypoxia or important in the cell's response to hypoxia, are there potentially ecological ramifications to these adaptations for the snow leopard? And so over the last 15 years or so, we've been collecting samples of scat, with colleagues across these different countries, right? And they're typically been based and then they've been done through just these non-invasive surveys to look at distribution. And then we've used these samples to look at population structure and phylogeography, to look at diet. And now we have this opportunity to use these same samples to start to look at genome variation across the range and to also start to look at these functional variants. And so what we did initially is we sequenced 5 snow leopards using Illumina, the entire genomes, for 5 snow leopards at 2x coverage and 1 snow leopard at 20x coverage. And we did this to try to characterize additional variants or SNPs that are present in the snow leopard genome. And we found that there was about 250,000 SNPs that we're able to characterize in this particular species. Now, these snow leopards were all captive individuals, right? So the next is, how are these SNPs distributed, and are they informative for looking at population structure in wild snow leopards? The other thing we wanted to do, again, is start to look at the functional variants that are basically present in this species and to look at the potential distribution of polymorphism in wild populations, right? And so we found these locations of these SNPs in those assemblies, and then the reference sequences that we assembled together, or we mapped to the references, I should say. And then we designed primers across, or I should say primers that were on either side of these different variants. And then we attached adapters that are basically enable us to then incorporate the barcodes that Illumina uses for multiplexing. We amplified these particular markers in the samples from the different areas, and then we cleaned them up with a bead cleanup. We did a second PCR where we attached a unique barcode to different samples, again cleaned them up, and then we put together a library using these amplicons that we sequenced on an Illumina instrument. And we initially just sequenced 10 different snow leopards from Mongolia, from Gurman-Saika National Park. So we did the sampling with Dr. Munkzak. And then we also sequenced 11 from Baltistan. And so these were samples that were collected by Dr. Shafqat Hussain and his colleagues. And what we found is that there actually was polymorphism at that EPAS1 gene. There were more homozygotes for the derived allele. So the derived allele is the allele that's unique to snow leopards. So there's a mutation in snow leopards at some point in time after they diverged from the tiger. And there were more homozygotes for this derived allele, which presumably would be the adaptive allele, the high altitude allele, than expected based on Hardy-Weinberg equilibrium. In addition, there were no ancestral homozygotes. So there were no snow leopards that were homozygous for the allele or the form that is present in the tiger. And finally, there were actually more derived homozygotes in the higher altitude region in Pakistan than in Mongolia, right? And so again, this is actually consistent with the fact that it is an adaptive beneficial allele at high altitude. Now, because there's still polymorphism, there's potential that the polymorphism allows snow leopards to occur in a lot of different elevational zones because they seem to still have the low altitude version and the high altitude version. And so we described some of this initial work, some of this preliminary work in the recent book chapter that has come out this year. Now, what are the implications for this? Well, first of all, snow leopards can occupy higher altitude regions than other carnivores. And again, is because they're partly adapted to the low levels of oxygen in those areas. By having polymorphism at this one important gene, EPAS1, they are potentially able to live in different elevational zones and at a greater amount of elevational different areas than other species potentially, right? Now, because there's polymorphism, that means that there's this potential for regional differences in tolerance to hypoxia, right? And again, this is preliminarily shown between Mongolia and Pakistan, where there's more high-altitude homozygotes, or for the presumed high-altitude candidate gene, the homozygotes for that particular allele, I should say, in Pakistan versus Mongolia. And there's more heterozygotes in Mongolia, which is a lower elevational area. Now, when you think about it in terms of conservation, well, why is that important? Well, first of all, you got to think that hypoxia is going to impact the competition between different species and different carnivores, right? So basically, presumably, in a certain elevational zone, the species that are better able to tolerate that low level of oxygen are going to do better. They might outcompete. Now, other important consequence is that when you think about it, like if we have these elevational changes in the habitat distribution or prey distribution, the thing is the amount that snow leopards can track these changes is going to basically depend on their physiological limitations, right? And so by understanding how these alleles are enabling them to live at different elevational zones is important in modeling the potential response that there's going to be. Now, there's also a lot of important challenges to incorporating NGS into wildlife analysis. Now, what we did initially was just kind of a preliminary analysis of incorporating NGS to genotype some important SNPs in snow leopard scat. And we can, of course, expand this out to try to extend how many SNPs we're doing, how many SNPs we're looking at, to try to get a finer look at population structure, for example, or potentially use it for identifying individuals. But by using NGS, we're basically creating extra challenges for incorporating genetics into wildlife studies, right? And some of these just simply, there's high upfront cost. It costs more to generate Illumina sequencing data. Not every lab is going to have access to these types of instruments. Also, the DNA quality and quantity has to be better for doing NGS sequencing than, for example, for doing microsatellite analysis just in general, right? Again, there's methods that you can use for degraded samples, but in general, it's better if you have good quality samples, right? The other problem is after you get the data, after you sequence it, for NGS applications, you typically need specialized computers and software. That costs more money. The data files are bigger. You need computers with more speed, more RAM, etc. Typically, for a lot of these software programs, you can't use laptop computers. You at least have to have a desktop. Sometimes you have to have specialized workstations. Sometimes you need the Linux operating system. Some of these things won't run on Windows. And some of this specialized software either is expensive, right? So there's these software that are easy to use. They have a nice visual interface. A lot of these are commercially provided, and so you have to pay for them. And then the ones that are free, they work great and they're available. However, they often require additional programming skills to actually use. They require being familiar with Linux. They require some coding experience, right? And so basically all this means is that for you to get into genetics using NGS, you typically need more training in the lab and more training in the analysis. The other problem that we have is that because there's so much data, you no longer can look at all your sequences and genotypes that you generate like you can with microsatellite analysis or traditional Sanger sequencing, right? So any kind of errors in contamination and sequencing, technical artifacts, they're more likely to get incorporated into your results. And so you have to be a lot more conscious of it, a lot more careful when you do the analysis and when you interpret the data. In addition, another challenge is that there really haven't, this is a developing field, right? So in some cases, there's not the software available or algorithms to incorporate all the data that you're generating. So for example, for functional analyses or for population structure, everything is focused on SNPs. For GWAS studies that look at the link between genotypes and phenotypes, people tend to focus on single nucleotide polymorphisms, right? But there's all this additional information on insertions and deletions, copy number differences, and structural variation on chromosomes that is generated when you sequence, but that data hasn't yet really been incorporated into a lot of these approaches to data analysis, right? And so again, there's kind of this lag. It's a really developing field, right? But it's going to have a lot of things. It can really open up a vast amount of questions that you can address. It's just that there's basically more more effort and resources that are required to do it. Right? And so there's a lot of resources that you can use to learn these different things and these different approaches. I teach a bioinformatics class at Duquesne University in the fall. This semester, I started a YouTube channel, Genomics, where I'm posting some of these training videos and informative videos and how to use different software, different bioinformatics tools online. I'm going to try to also post lab videos and lab training videos for genetics, such as DNA extraction, PCR, and so forth. There's other similar videos that are available on YouTube. I really encourage you to, just try to get your experience with, doing something hands-on. So again, try to volunteer or work in a lab so that you can learn some of these skills and then apply them to wildlife and to snow leopards. And so I would like to thank everyone that I work with. It's a really long list. It's been amazing to work in the snow leopard field, interact with everybody. It was really nice to participate in this workshop. I really enjoyed meeting you all via Zoom. If you have any questions or if you have any things that you want to discuss, please feel free to contact me. And again, I look forward to hearing from you and hopefully meeting you sometime out in the snow leopard habitat in the mountains. Thank you.